I'm here to talk about my research, which involves a lot of the applications we've talked about today with whey, specifically WPPC, and some of its nutritional applications to mice, specifically whey protein phospholipid concentrate fractions and how they can enhance bone density and microstructure in mice. I'm a big believer of starting with why, so I want to talk about why this research is relevant to me and the world around us. And we live in a demographic of shifting older populations. As these demographics are aging, the world population is going to be significantly more. Sorry, I lost my train of thought there. As these demographics are shifting, the population of those aged 65 and above is only going to increase more and more over the next decade. Because of this, as these older populations become a larger part of the demographic, age-related chronic diseases are going to become more and more prevalent. Specifically, osteoporosis and sarcopenia are two that I picked for my research. Osteoporosis and sarcopenia are both closely related diseases. The coin term osteosarcopenia has often been used. The prevalence and severity of one disease can often impact the prevalence and severity of the other. And then kind of the oddball here, which is an underutilized dairy co-product, whey protein phospholipid concentrate, which we've talked about earlier. My research is bringing all three of these together and looking for ways we can use WPPC as a nutritional solution to hopefully alleviate some of these age-related chronic diseases. I want to talk a little bit about nutrition and how important it is for bone health and developmental health. It's well known that nutrition has a huge factor to play in bone health. Everything from not just the calcium you have, but the bioavailability of the calcium, vitamin D, magnesium, other vitamins, and protein and fat sources. It's also well known that dairy sources are among some of the best in terms of nutritional sources to support bone health. I also want to talk a little bit about bones and how they develop. So as a developing bone, laser pointer, we're not going to use that. As a developing bone develops, the osteoblasts, or the bone builders, build the outward bone and remodel some of the internal structure. And as the bone is developing and growing and expanding, osteoclasts are responsible for remodeling the inside of the bone and redistributing some of the calcium elsewhere. So the balance and equilibrium between these two types of cells is essential for building healthy bones. Whey protein phospholipid concentrate is produced in huge abundances. We've talked about how, Scott talked about how there's so much whey produced in the world. A lot of that whey is processed into whey protein isolate. This is the high valuable commodity that a lot of people are looking for. It's used by bodybuilders. It's very popular, high value. And 140 million pounds was produced in 2023 in the US alone. With this 140 million pounds, that means kind of kitchen table math, or napkin math, about 75 million pounds of WPPC was made on a dry basis. The demand for WPPC is much lower than WPI, and because of that huge demand for WPI, WPPC just keeps getting produced and produced, and it doesn't have a huge effective use right now. Most of it is really used for animal feed. Very little is actually used for human consumption. There are some niche applications in use as a dairy supplement or for infant formula, but those applications are growing, and the demand for WPI and the resulting production of WPPC still far outweighs the demand in these kind of niche products. We've seen this figure before a couple of times, so I don't need to harp on it too much, but I do want to point out one key step. So the cream separation step at the top, when you separate that wet whey and you separate the fat out, the residual fat globules are smaller in size. And when you go through the whole processing step down into the retentate into the final WPPC at the end, those small fat globules remain in that WPPC. And because of this, we have a lot higher surface area to volume ratio of these milk fat globule membranes and these milk fat globules. The triglycerides cores are smaller because they're smaller milk fat globules, resulting in an enriched milk fat globule membrane in WPPC. And we've talked about this a little bit before. You've also seen this image. So I'll go a little bit quicker through it. But in WPPC, we characterize these milk fat globule membrane proteins, or MFGM proteins, as bioactive and very valuable. About 22% of the protein in WPPC are these MFGM proteins. These proteins can be enriched to 5, 7, 8% of the total protein, which is much, much higher than any of the other available dairy products or precursor products in this dairy stream. Also, the phospholipid distribution is unique because of the higher enrichment of that phospholipid membrane of the milk fat globule. Coming back to the osteoporosis side and sarcopenia, we think WPBC may provide a nutritional solution, so kind of connecting these two ideas together. There was previous work done by my professor, Dr. Osterk, during her postdoc time at UC Davis, where they took WPBC and they introduced it to weanling mice diets. They saw in both a high-fat diet and a low or normal fat diet that they had significantly longer femur length. So an interesting bone phenotype occurred when they introduced WPBC into the diet of mice. The key factor that my project was aimed to try to uncover is WPBC itself is about 50 to 60% protein, 30% fat, roughly. Different producers have different ratios of this. And we weren't sure if it's the protein or the fat that could be causing this bone phenotype. So that kind of leads right into the next slide. It's almost like I designed it that way, where we fractionated WPBC into its lipid and protein components. This is actually the same product that Sonali used, the UF retentate, or our enriched protein fraction here, and then our enriched lipid fraction on the side. We used a filtration approach, like Sonali said, and then a centrifugation treatment approach to produce our protein and lipid fractions from our original WPBC precursor. As you can see, we were able to enrich these to relatively high content, almost pure protein, almost pure fat, and then we introduced this into mouse diets. To look at the composition of these, this was also already shown, so I'm going to go a little quick through it. We see a lower abundance of milk fat globule membrane proteins in our produced protein fraction. The relative distribution of the MFGM proteins still is there, so there's still MFGM proteins and the relative distribution is similar to WPBC, but the total abundance of MFG proteins is less. And we see the enrichment of those predominant milk proteins like lipocalins such as beta-lactoglobulin in our protein fraction compared to WPBC. So the produced protein fraction we made is similar to but distinct from WPBC itself. It's unexpected results from the membrane filtration method that we use to create the protein fraction. With the lipid profile, we see similar results where the overall distribution of many of these unique phospholipids and fats persisted in our extracted lipid fraction, but we did see an artificial enrichment of triglycerides as a total fraction of the WPBC lipid fraction that we produced for the mice. Going into the experimental design, so I think this is really key. The diets we designed for the mice were isocaloric in nature, meaning they had the same amount of protein, the same amount of fat, fiber, carbohydrates, and even vitamins and minerals. We broke the mice into 4 separate groups. So we had our control diet, we had our protein diet, which had the protein fraction added at 10%, our fat diet with the fat added at 10%, and our WPBC control as a positive control where it was whole WPBC added at 10%. We had an N of 9 for each group, total N of 36. And it was a 13-week study involving weekly body weight measurements, biweekly body composition scans, and 4 total X-ray scans taken throughout the course of the study. And then we've got a nice little picture of my cute mice in their cages. They were very fun. It was my first time using mice. Quite the experience. This is a really ugly slide, but I want to highlight 2 really key things. So this is the total diet formulation for the 4 diets themselves. In order to balance the diets to make them isocaloric, we had to add whey protein isolate as our protein source to the non-protein diet, and we added lard to the non-fat added diets. This was done intentionally to use protein and fat sources that were most similar to WPBC, as in animal sources, or literally whey protein isolate, the other side of that final filtration phase, to try to avoid any confounding variables from using non-plant or non-animal-based proteins and fats. There was also an excess of vitamins and minerals added, so that if there were any small differences in minerals, it was negligible due to the excess of vitamins and minerals added. When we look at the body fat, body lean mass, and body weight over the course of the study, we don't see any significant differences. So the isocaloric diets in nature, this is probably an expected result, where the body fat, the body weight wasn't really changed. All of the mice in all groups were able to grow up and grow in a healthy manner. And it really serves to highlight some of the other differences we do see. So we can say that some of the other differences aren't necessarily due to changes in body weight or body fat. When we look at the X-ray scans, so the 4 X-ray scans taken throughout the study, I've got a representative image on top showing how we measured the femur length, and then the X-ray scans that looked at bone mineral density. I first want to talk about the femur length. So by week 12, we saw significant increases in the protein group and WPPC in femur length of the mice. So body fat, body composition all stayed the same, but we're having longer femurs with our protein and WPPC group. So not only was this a significant finding, but it also models the previous work done by my professor at UC Davis, where they found a longer femur length in their weanling mice as well. When we look at bone mineral density, we see that there's a significant increase in weeks 8 and 12 in bone mineral density for the fat group. So the protein seems to be doing something with femur length, and the fat group seems to do something with bone mineral density. And we see the same trend reflected in whole body bone mineral density. So WPC protein may be associated with an increase in femur length, and the fat source from WPPC seems to be associated with an increase in bone mineral density. When we wanted to look into this phenotype more, we conducted micro-CT scans. So post-mortem, we harvested the femurs from each mouse, and we did micro-CT scans to look at the internal microstructure of the bone, looking at that trabecular and cortical bone. As you see on the representative figure on the left, we had 2 regions of interest that we looked at. The midpoint of the bone, which is ideal for looking at the cortical bone structure where there's not much internal trabecular bone, and then the distal end of the femur kind of by the kneecap, where you have very dynamic and complicated trabecular structure to look at and analyze. When we look at the cortical region of interest, we see that cortical thickness for all groups was qualitatively increased and significantly increased for WPPC. So qualitatively, all the groups have thicker cortical bones, significantly with WPPC, and then protein and fat were also trending towards significance, but didn't quite make the p-value cutoff. We see another very similar story with the cortical bone mineral density. So instead of a 2-dimensional analysis, this is a 3-dimensional analysis of the bone mineral density of that cortical bone itself. And we see, again, that same story of enhanced cortical or higher bone mineral density in this region of interest for our experimental groups. Looking at trabecular structure, we have the trabecular bone mineral density, or the density of those internal trabeculae. We have trabecular thickness, or the literal thickness of these complicated internal structures, and then the average trabecular number, as well as the spacing between the trabeculae. An increase in all of these except trabecular spacing would be associated with an increased bone health. We can see with bone mineral density, qualitatively, all groups are higher with significant increases for fat, which correlates very well with our 2-dimensional X-ray scans from previous slides. And then we also see an increase in trabecular thickness for both the protein and the WPPC group. So thicker trabeculae inside of the bone. With trabecular number, we don't see any significant differences between groups, so the number of trabecular are similar inside of the bone, but the thickness and density are higher, as well as the trabecular spacing being lower. So when we kind of put all this into an image in our minds, which I like to do sometimes, we have thicker, denser, and tighter packed trabeculae inside of these bones of the mice, just from a small change to the mouse diet. We wanted to look at the possible mechanisms that this could be causing. Obviously, we have a pretty significant phenotype here with our mice, and we wanted to look at whether or not the transport of calcium across the gut could be associated with this increase in bone health. In order to do this, we looked at some key, we did PCR of some key genes that are in the ileum and jejunum of the mouse. Specifically, we sampled the jejunal mucosa. TRPV6 is an important calcium transporter in the gut, and S100G is an important calcium binding protein in the gut. We didn't see any changes in these, so right now our hypothesis is not necessarily leading towards the idea that the mice are able to uptake more bone into their bloodstream. But we also looked at FGF23 and BMP1. These are more bone regulatory proteins that have some expression in the jejunum. We see a small change in FGF23, which is associated with vitamin D and parathyroid hormone, and no changes in bone morphologic protein 1. So right now, our hypothesis is not necessarily pointing directly at the idea of increased calcium uptake. We think it's more along the idea of gene regulation in the bone itself, or that equilibrium between osteoblasts and osteoclasts. We have some ongoing work right now to further explore this mechanism. So we have some 16S analysis of the microbiome, kind of with Sonali's work, where they observed different changes in microbiome with the addition of the UF retentate, or the protein fraction that I used. It's the same substance. So we're trying to explore if the changes in microbiome could be changing gene regulation and affecting the bone itself. And we're also doing RNA-seq of the L5 vertebra, which is a great candidate for bone regulation, to look if there's any to do an untargeted analysis and see if there's any upregulated or downregulated genes that we can kind of catch and look more into. We're also doing PCR-specific bone genes that are associated with osteoblast and osteoclast regulation. So our future steps and the conclusions I want everyone to take away. I know it was kind of a dense presentation, but WPBC and its fractions can enhance bone mineral density and internal femur structure. WPBC-derived fractions also have different effects. So we noticed that the proteins seem to have something more to do with femur length, whereas the fat from WPBC seem to have an increase in bone mineral density. And all of these differences were a result of a very small alteration to the diet of the mouse. Only a 10% change to the mouse diet resulted in these significantly detectable differences in healthy mice. These were not diseased mice. These were mice that were already healthy, and the addition of this small change to the diet made their bones even stronger. The underlying mechanism, we suspect it to be related to calcium uptake in the bone rather than the gut itself, and we're looking at that osteoblast-osteoclast equilibrium I alluded to earlier. And we also plan to explore the effect of WPBC in osteoporosis and sarcopenia in a post-menopausal mouse model. So moving from that healthy mouse model that we've observed significant changes in already to a diseased mouse model, where we'll look at the possible rate of bone loss in an osteoporotic mouse. I'd like to acknowledge a lot of people, because this was a pretty big project. A lot of these people are in the rooms, and if you ask any super detailed nutrition questions, I may direct your question to them, so people out there be ready. Definitely my lab for sure. The Pierre lab has been absolutely huge in helping me. The Meyer lab, the Crenshaw lab, and then Mike Molitor and the CDR team have been instrumental in helping create these diets. Definitely couldn't have done it without them. And then got to plug the DIH for funding my research. They have made all of this possible. Thank you.